So I deliberately added an S to the title here, because what we're going to be talking about today, I think, is an approach to the biggest challenge, which Charles will also be talking about, how our choice of projects and target validation ultimately drives the underlying abysmal cost model which we have in the pharmaceutical industry. We faced an position with some of the more recent developments where it's potentially several billions of dollars is the cost of bringing a drug to market. And that's not the individual cost for any one project. It's the cost of failure for when most of our projects fall down. So one of the things we need to start thinking about is to challenge some of our underlying assumptions of how we do in drug discovery, and actually is it a philosophical approach which is driving some of this failure. So over the past few years, we've been trying to sort of go through the looking glass and whatever, come out the other side of a rabbit hole, and we're trying to find out, is there another way to think about this? And it sort of goes back to a quote by Marcel Proust that discovery sometimes doesn't necessarily consist of looking for new landscapes, but actually new eyes in how we're trying to see it. So this led us then to thinking about how then we could actually look at the actual problem and think of a new way to approach it. So one of the underlying, I think, lessons that the drug hunter needs to learn from all the work we've done over the past 15 years in the human genome and functional genomics is actually the sheer degree of degeneracy, redundancy, and robustness of biological systems. Ultimately, what we're trying to do is perturb a phenotype. And in fact, many of our problems fail, not because they don't necessarily work efficacy in phase 2 or phase 3. They often fail because they just about don't work, or they don't quite meet the standard of care. So it isn't as if we drive in projects into a clinic where there is no disease hypothesis. We often got a lot of data to do, but they just haven't quite got the horsepower to drive it through. And this then sort of tantalizing problem actually also blinds us potentially what's going on. So a real challenge is how do you come up with a drug that's better than standard of care and potentially is going to perturb the phenotype. And of course, many of the exciting work we're seeing now is that the underlying biological systems and the network structure actually drives and is responsible for this degree of redundancy and biological robustness. A good example of this actually to show how we can expand this opportunity space is of course what we're seeing now with what we call approaches to synthetic behaviors, where it's synthetic lethality, where knocking out one gene has no effect, knocking out a second gene has no effect, but together there's an emergent phenotype which potentially is lethal. And people like Steve Jackson in Cambridge this year have published in Science where they've also shown synthetic rescue against rare diseases, where one perturbation is the rare disease mutation, but potentially by a second perturbation of a small molecule chemical against a previously potentially unexpected target can actually rise to rescue that phenotype. So this then is a way then of thinking about how do we expand our opportunity space for new pharmacologies? And this then, we think, is we take a sort of a multinodal or at least a binodal approach to it. We can expand opportunity space. So one example of this was a paper from Toronto in Science back in 2008. And it's just a nice example in yeast. What we did is take each of the individual gene knockouts. About 19% of all genes in yeast are lethal when they had a single perturbation. But then they added a second perturbation by screening each of the knockouts against a wide variety of different chemical tools. And then by having two perturbations, a genetic perturbation and a chemical perturbation, the potential number of lethal genes observed then rose up to about 97%. So it's a 5-fold increase on potentially what we thought was unimportant actually turns out to be important. And this one is a nice analogy to thinking about how then we could start thinking about expanding in certain areas potentially the pharmaceutical opportunity that we may have by potentially mixing and matching targets. And a really nice sort of explanation of this was in Nature Reviews Drug Discovery, Scannell et al., a couple of years back now. And it's a lovely quotation, and it's an analogy I want you to go away thinking about. So what they say is that many drugs derive a therapeutic benefit from interactions against multiple proteins rather than single targets. When we look at many of those small molecule drugs, this is absolutely true, far less specific than biologics. These drugs are magic shotguns rather than magic bullets, and that's a phrase Brian Roth coined. If this turns out to be more generally true, then worrying about the low-hanging fruit problem, have we got enough druggable targets to go after, would be similar to worrying about the shortage of notes threatening the future of music composition. We only have 8 notes, but we can put them together in almost infinite variety to create music. And again, with potentially with drug targets, how they are blended and put together, almost like creating chords, is a way then to think about the emergent pharmacologies which could come out from this. So one of our sort of underlying philosophies behind this is to start off with the assumption that the clinical efficacy and safety profile of a drug is not determined simply by the target we're trying to aim for, but ultimately, of course, by its activity profile across multiple proteins in a proteome. What we would call its polypharmacology profile. And if we start off with that as our assumption, then we need to think about sort of design methodologies that would allow us to rationally try to design against profiles, not just individual targets. We might want to bring in additional targets to increase the efficacy, or importantly, of course, we want to design out the anti-targets and consider these a priori to improve safety. And the way we think about doing this is to look at it as a computational approach, is to think about this as a complex problem we're trying to solve in trying to define and try to find chemical matter for complex target product profiles. And we've been developing a variety of techniques which combine things like evolutionary computing with the wealth of large data sets which are now available. Since we started doing this, we first got into a field when we started to work with people like Informatica, which then led on to become the Chemical Group. And when we started doing that in Pfizer and sort of published our first sort of views on pharmacological space in Nature in biotechnology, we started to realize that we just weren't exploiting the huge amount of historical precedence there is for medicinal chemistry. For example, you can use all of the sort of the large data that's now available of all our pharmacological projects, whether public, in patents, or in your proprietary data sets, to think about how you would mine out particular medchem tactics, how you'd use this data to derive various different machine learning models to predict potential pharmacologies. And of course, the work of Brian Shoykat in UCSF and our group about this ability to try to predict then using this large amount of data. So this then for us has been one of the breakthroughs along with cloud computing that allows us to scale up these kind of algorithms. Just to give you an example then of what I mean by trying to design automatically against a sort of profile of drug targets. I'm going to take you through a few simple examples which we recently published in Nature and then show you how then this could be exploited on a more sort of commercial sort of basis. So we always like to learn from the greats, and Sir James Black once said that the best way to discover a new drug is to start off with an old drug. So here we start off with Aricept, donzopyl acetylcholesterol inhibitor, and still, tragically, one of the few compounds approved for the treatment of Alzheimer's in early-stage cognitive enhancement. And interestingly enough, we predicted it potentially to be, have additional activity against dopamine D4, and when we measured it, certainly does, 600 nanomolar against dopamine D4, as well as being active against acetylcholinesterase. So it's just an aside, and it's very interesting that a drug that is one of the few drugs approved for cognitive enhancement actually hits two targets, both linked independently potentially to cognitive enhancement, acetylcholinesterase and dopamine D4, which has been linked to that for a long time in literature. So what we want to do now is just take donzopyl and see if we can evolve different activities. And what we're going to show you now, we're going to show you some more detail, but this is what we're trying to do. You mine out a variety of different ways and transformations, which we learned from how med chemists think about designing drugs, and you profile these against a wide variety of, say, 20 different GPCRs here, and we're trying to drive them towards individual specific profiles to make sure that our predicted polypharmacology profile potentially matches. And this can either be driving it towards a single target or driving it potentially towards multiple targets. The maths is the same. So some slides I borrowed from a friend of mine in the audience, Larry, which tries to sort of summarize the sort of projects we were looking at. So one starts off with Aricept. Then, before you walk before we can run, as you say, just optimize it against dopamine D2 and make sure this is an oral CNS drug. We then evolved the compounds and came up with this particular chemotype here, which we made 7 compounds. In doing so then, you realize that we start off with something that's basically inactive against D2, and now this particular series of compound, this one is particularly 156 nanomolar. So we've evolved and created that activity within this compound. But all of these other targets have activity, so all of these come along from a ride because we didn't actually select against them. And this is very common if you're just picking a compound from HTS hit, might have derived activity, but also has a lot of liabilities. Particularly here, we see that the alpha receptors are actually picomolar and far more potent than the actual dopamine receptors. What we do then is take some of these compounds here. They become a starting point for the next evolutions. Now we change the objectives. This time we say we want it to be so novel we could file a patent, get into past the blood-brain barrier. All of these are measured in mice and certainly do that. But this time, we also want to have a multi-target profile against 5-HT1A, the dopamine D2 receptors, but absolutely improve the selectivity of the alpha-1. So typical sort of anti-target reduction problem you'd have in medicinal chemistry. We've run the algorithm again, and this set of benzolactams certainly now are much weaker, 300-fold more selective against the alpha-1 receptors, but we've also maintained nanomolar potency against the two targets we wanted. So this then sort of gives us sort of some examples to show how a computational algorithmic approach can be applied to some of these design problems we're trying to solve here. We then took a sort of another set of examples where we then wanted to optimize this further, and we then take donzapil again and have evolved this then further just against D4, and we now see that we went from 600 nanomolar now to a compound which is about 9 nanomolar. Now, this was an interesting compound, but not necessarily patentable in its own right. The chemotype had been seen before. So now we put much more stricter definitions. We want to be highly selective against dopamine D4, and we're going to evolve it now against the CNS. Everything else becomes the anti-target. We evolve that further, and there we go. It's now 90 nanomolar against it, but highly clean, this particular chemotype of morpholinos against its target. What was interesting about this is that all of this sort of med-chem work was done by the algorithm. We filed a patent on this series then just to prove a principle that intellectual property can be generated algorithmically and therefore also fulfill some of the sort of common hit criteria one would have below 100 nanomolar, selective for the series, et cetera, et cetera. Overall, the entire process was about 40-odd compounds to synthesize, tested in a matrix of 20 targets, and what we see was a very high congruence, about 75% of the predictions, whether or not it hits a particular GPCR or not, are proved correct by this sort of methodology, which gives us confidence then, if you can start predicting the profiles, you can start to design against particular profiles using such sort of automated techniques. We were then lucky enough then to create a sort of spin-out company after publishing a paper to see if we could take us further onto some more challenging approaches from the pharmaceutical industry. We formed a company, Excentia. Company's doing rather well at the moment. We've got deals with 4 companies looking at a range of projects, particularly sort of in the biospecific design area, which I'll explain in a few minutes. But it's also given us real market validation of whether or not this approach actually can be fruitful. So one of the things we're really interested in is how do we then use this kind of technology to try to answer our first question, which is how do you potentially enhance efficacy? So we're coming back to the proposition that many existing drugs potentially do target several different receptors, and in several disease areas, that is a beneficial thing to try to achieve. But of course, we do this in many other disease areas because we can't do it with individual drugs, we have to do it with combination therapy. But the limitations we have here is that we have to balance certain half-lives and the PK and side effects profiles to get the right balance. There's potentially a superior approach that could be developed by using it in a single agent, a bispecific agent. And some of the same philosophies have been applied to the development of bispecific antibodies, potentially also apply to this philosophy as well for small molecules. What I'm going to show now is, unfortunately, I have to anonymize it because it's not yet patented. A particular collaboration with a pharmaceutical target company. I won't fully disclose the targets or the compounds, but you will see a gist of what we're trying to do here. So this particular one, we were interested in the challenge of a new class of anti-diabetic agents, try to improve standard of care. And the design objectives are first-in-class profile. The 2 targets, the 2 enzymes, are completely unrelated. So previously we were working on GPCRs. Now we're applying it to a structure-based approach where you're taking 2 enzymes which have no similarity in terms of their underlying substrates or in underlying enzymatic activities. And of course, you want to be selective over other members of a gene family. You don't want it to be promiscuous against many of the targets. There's about 10 or 20 other members of each gene family, selective over those, and of course, patentable. And what we do then is that we define, starting off with some of the large databases like eMolecules or the Chembl database, and you triage through those and evolve in the compounds for generations and generations to define the set of objectives one is trying to aim towards, and then a set of anti-targets one is trying to avoid, until one gets to a point where you think you've potentially reached those within a few generations. And then we make just a handful of compounds. Usually when we test on the hypothesis, it's between 5 and 20 sort of compounds are used to test a hypothesis. What we mean by hypothesis is that a particular chemotype is likely to have a particular pharmacological profile that one is looking for. These compounds then were synthesized, a handful of them, a few false positives against one of the enzymes. This is an interesting compound by itself, just for this particular enzyme, being highly selective. But then we find that actually the number one compound which the algorithm told us to synthesize is actually 300 nanomolar against one of the enzymes and 11 nanomolar against the other one. We then were lucky enough for our collaborators to then go to solve the crystal structures of each of the different enzymes, then both bind in the same sort of top hit, and this then allowed us to confirm that our structure-based approach hypothesis was correct. And so this is a very interesting way now we can think about how to optimize this further. Now we have both crystal structures to hand as well. The other thing as well we could be thinking about, if we could think about multiple targets, is by really biting a bullet of where we could actually take target discovery in academia. So one of the key things we've been thinking about as well is obviously an important response to the emerging new interest in phenotypic screening that is coming about. And one of the issues we face in academia is that it's some fantastic biology, but it actually is potentially a long way from being implemented and turned into industrial scale assays within the pharmaceutical industry. So we wanted to see if we could try to then create a new sort of conduit between working with academia and actually then producing it in an industrial scale, high quality approach to see if we could discover new lead compounds and through target deconvolution, potentially new targets, by perturbing now the wealth of patient-derived material and stem cell material, which is currently being funded, particularly in the UK and Europe. And actually this goes back to another piece of wisdom from James Black. So he published a very interesting little article after he won the Nobel Prize, outlining his wisdom of what you should think about if you're going to run a drug discovery project. So there's 6 rules. Is a project purged of wishful thinking? Is there a chemical starting point identified? Are there relevant bioassays available? Will it be possible to confirm laboratory specification in humans? Is a clinical condition relevant to item 4? And does the project have a champion? So what's interesting about 3 out of the 6 of these highlighted in orange are all to do with how translatable your assay is in representing the human disease in the laboratory. So he really is hammering home the same message here again and again. Does your assay represent the potential disease? So this is where we think there's a role now in how do you link with work that's going on in academia, potentially then to thinking about how we start converting these and translating them into hopefully assays that are ready for larger scale discovery operations. And of course, what we're talking about here is potentially on this scale is that we've done a huge amount of work in this space. And there's probably at least 2,000 plus targets which we've run HDSs for and other lead discovery campaigns in the industry over the past 30 years or so. But what we want to move towards is potentially more disease-relevant approaches, where it's more complex cell models. There's a few interesting talks this afternoon and tomorrow how to do that with 3D systems and co-complex systems to move towards disease models and even potentially tissue systems as well, which some companies want to go straight into screening now due to the disease relevance. So what we're talking about potentially is how we start engaging and learning together about how to conduct these sort of this science, but actually in an open access and open source way, much in the same inspiration that the SGC has tackled the whole problem of high-throughput crystallography and chemical probes and actually solved that problem. So my own interest in this area came through a project we started to do from a sort of informatics point of view with Quentin Bittel and a project funded by the Gates Foundation, trying to create a phenotypic high-throughput screen for schistosoma larvae. What was interesting about this is a neglected disease. And the assay was being run literally by looking down a microscope and counting how many particular larvae were dying under the drug treatment. What we realized, of course, is that with modern image-based informatics approaches and machine vision, one could potentially develop a high-throughput approach to this, of course, by developing algorithms which recognize the particular individual cystosomes, and importantly, running this over time-lapse feature so we come back to the same organism within a few minutes to see how its motility and morphology are changing. We did this in collaboration and also with Biovia, who also previously were known as Accelerus. So the beauty of this approach is that we can start then to, forgive the misspelling of Bayes, is that we can start to think about how then we could start to classify phenotypes by the wide range of high content features which we got from this. As in most high content models, you're dealing with potentially a few hundred features. These then could be used to build sort of various models to start to classify the different drug responses. Here we've got a phenotypic model, and here we have a motility model that was built. And what we find then, is we elegantly start to classify out the different drug responses by these sort of approaches. This then allows us then to start, when thinking to screening a new compound, not just whether it lives or dies, it kills the organism, but potentially what sort of drug-induced phenotype it has as well, potentially, by looking at a similar phenotype. What we tend to do then is that recently we've been able to raise sort of a large amount of capital money, about equivalent to about $14 million, just to spend on sort of capital equipment, to put together a collaboration between in Dundee and Edinburgh in Scotland with the University of Oxford, how then to build, we call it a UK national resource, but absolutely it's completely open for business for transnational collaborations across the world. And that's one of the beauties of some of the private funding we're trying to raise behind this. The whole point of this really is how do we take some of the new approaches in engineering stem cell approaches, whether it's CRISPR, whether it's patient-derived cells, and use these then to try to create very difficult assays which hopefully will be much more disease relevant than conventionally we've been developing. And the idea behind this actually is potentially to be quite disease agnostic and to therefore work with this technology platform then is hopefully designed to work with a wide variety of different academics. One of the philosophies we'd had was not to develop screens to fit your technology, but try to fit your technology to a wide range of screens available. So that means we've gone for a wide variety of different readers, using high-res bio-robotic systems. And this then allows us then to be completely flexible in how we want to think about developing the assays, whether we look at a confocal or wide field, where we're looking at some of the label-free approaches which are available to us now. And with the assay development skills that were reflected within Dundee, Oxford, and Edinburgh, including some of the one of the ex-heads of AZ's high-content screening facilities as well on board, hopefully would allow us then the flexibility to be very creative in how we want to start thinking about developing assays here. One of the ways we want to think about it, actually, is we are developing the informatics approaches, which would allow us to think about how we do the screening live on the fly. As we collect the data, how do we sort of try to solve this problem? Is that we have relatively low throughput assays, but at the same time, we want to explore both biological and chemical diversity in the compounds. And this has been one of the challenges previously. But what's interesting is that our approach is now to thinking about how do you do adaptive, iterative screening, which actually the screen tries to learn as it runs and collects data, rather than just take a whole bunch of compounds and screen through them one to n. So this is built on work that was done in Pfizer originally, where they started off, rather than screening 2 million compounds, they discovered that they took certain diversity sets that represented that larger space, then saw some initial signals from that, and then selected out the next plates with the highest likelihood that they could potentially find 90 to 95% of the hits within 50,000 compounds, as they would by screening all 2 million. And we believe a similar sort of philosophy can be applied in this space as well. People like Ross King in Manchester are developing this kind of approach in their robot screening approaches. And this then would allow us then to try to then solve this problem of trying to explore chemical diversity within the sort of costs and limitations of these more lower throughput phenotypic assays. Of course, the other big challenge that we're working on right now, and we're in discussions with several companies, is how to put together the best possible set of chemical toolboxes that would also be biologically diverse. As I said, we've probably done lead discovery projects for maybe 1,000 to 2,000 proteins within inside of various companies. If we can take various examples of chemotypes from each one of those and then allow us then to perturb it in certain ways, and of course it gives us an instant hypothesis to go test if one of those annotated chemical tools goes this way. And Charles will explain to you, even in academia, the cost and difficulties of creating new chemical tools. So we should indeed try to make sure we can actively access and provide all the existing work that's previously been done to be available to biologists. The big thing, of course, that'll come out of this, we're hoping, is the ability of doing target deconvolution. How then, when you've got a phenotypic hit, does one use potentially genetic and biological approaches and biophysical approaches to work out what the actual mode of action our target is? But there's another way of thinking about this, and this leads back to the work we were doing previously on the algorithmic approaches. How, in certain situations, might one want to think about taking, high dimensionality phenotypic data, and could one potentially evolve compounds and design compounds automatically against that data without actually knowing a target? And this is actually an active piece of work right now, We aren't going to need direct results I can show you yet, but some of the compounds that we've already been thinking about hopefully will be tested shortly. But what it allows us to do then is to start thinking of a conceptual way how we can start to approach this. So of course, with all the chemical structures we have available, one of course can build various sort of fingerprints about how those chemical structures fit, of course, with the phenotypic fingerprints features. And the work we've done ourselves and other people, one can show then one can potentially build both models that classify the fingerprints, but potentially, if there's enough data, also models will start to link chemical fingerprints to phenotypic fingerprints. The other side of the equation, of course, each of these chemical structures, we have also the polypharmacology profiles, whether it's the predicted profiles I showed earlier, or whether or not one has actually screened these against whole panels of kinases or GPCRs or whatever targets one is interested in, and uses real phenotypic sort of profiling data. This is interesting, of course, because even if it is just a predicted target, it actually also becomes a surrogate fingerprint, which by itself potentially then becomes an object, which can an objective, which one can then think about designing against. So whether or not one has got a potential fingerprint-based model, or one has got potentially a polypharmacology profile, as we showed earlier, one then can think about using this data to try to drive such design algorithms both ways. And I'm hoping in a couple of years' time we might be able to come back here and present to you with some ways how we think actually driving this might be done. We think this is a really interesting way that we know some people are actively working in this space. But again, it's another way then to be driven by the pharmacological features whilst we're even waiting for those target deconvolution studies to read through. And I really want to thank the collaborators of Excentia. If it wasn't for working with industry partners, we wouldn't have been able to really develop the technology we have, and particularly Jeremy and Richard in my group. Really want to thank Brian Roth's lab, without who we wouldn't have ever got to this stage, Andy and Gilbert in Dundee. And also thanks to Sir Peter Radcliffe at Oxford and Paul Andrews at Dundee, who've really been instrumental in really driving the creation of this, hopefully this wonderful phenotypic screening center will allow a new relationship between industry and academia, particularly in that space. And thank you for your time.